next speaker. And this will be Dr. Mariarosa Mazza. She's a research fellow in nanomedicine at the University of Manchester in the Division of Cell Matrix Biology and Regenerative Medicine. And she will be talking about near-infrared nanoprobes for tracking of human mesenchymal stromal cells after implantation. Mariarosa is a pharmacologist by background, and it's all yours.
Thank you. Thank you very much for the opportunity and to the audience to come and listen to my talk. This afternoon, I will talk to you about one of the projects that I'm involved, which is the development of near-infrared nanoprobes for tracking of human mesenchymal stem cell after implantation. So, as Paolo was mentioning, in reality, my background is a pharmaceutical scientist, so how do I arrive here? In reality, is to try to develop a tool to help the development of cell-based therapy. In the case of mesenchymal stem cell, we know there are 59 clinical trials involving the use of MSC, because they can differentiate in specialized various type of cell, and for inflammatory, osteoarticular, and cardiac diseases. However, the performance of the preclinical success and the real clinical trial success of this cell-based therapy is hindered because we have a limited understanding of the fate of the cells once they get administered, once they get transplanted into the patients. So within our lab, we thought to try to use nanomedicine to provide tools to explore in vivo cell fate. And the hypothesis of this study is that the labeling of human mesenchymal stem cell before transplantation could enable us to track the cells after the administration, and therefore can be used as a tool for the assessment of cell-based therapies. Our aims is then to achieve a long-lasting effect of human mesenchymal stem cell tracking in vivo without having to influence the cell function and trying to eliminate the need for injection of contrast agents or genetic cell modification, which is a powerful tool when we are in the in vitro settings, but obviously is less appealing when we want to look at translation of the use of these cells. Therefore, we thought of a system that can encapsulate an ICG dye, which is an FDA-approved dye that works in the near-infrared region, and therefore can be exploited to look at the signal of the dye deep in the tissues in the body. And we have encapsulated the dye into a liposome using a composition of DOTAP-called liposome, and the classical lipid film hydration, followed by sonication. We can see from the 10 micrograph that we are able to form the classical unilamellar liposomes, and our repeats show that the system is quite reproducible. We have a hydrodynamic diameter of about 100 nanometer, positive zeta potential, and then we started to look at the encapsulation, at the incorporation actually of the dye. We can see that in terms of optical density, at a fixed concentration of dye, we have that the liposome ICG has a better optical density in comparison of the dye alone. And then we compared to a conventional dye that is used for labeling cell membrane, which is DIR, and however is highly lipophilic. And in the long term, we can see that while the optical density of the ICG decreases easily because it is known that ICG is unstable in buffers and aqueous solution as it forms oligomers and so on at high concentration, in the formulation with the liposome was highly stable. Therefore, we have taken our formulation, characterized, and we have a liposome in the 100 nanometer size range, positively charged, able to load 80 micromolar of ICG. Now, these are the dye that we wanted to compare the performance of ICG to the conventional DIR. And we see that when we put this into the cell media or biological buffers, we can see that the optical density of the DIR and of the dye-alone ICG is really poor when compared to the formulation of liposome ICG. And this also happens when we start to put it into cell media, when we want to progress for cell labeling purposes. So definitely the liposome ICG formulation outperformed the naked dyes. So we started to try to label various cell types, and we were able to efficiently label different type of cancer cell line. But also by doing this type of experiments, this is specifically New 87, what we started to notice was the difference in performance compared to the commercial dye ICG and the liposome ICG. So the DIR dye, which is the conventional dye that stains the cell membranes, was effectively staining the cell membranes, but the signal over time was decreasing very fast compared to the performance of the liposome ICG that was, we were able to pick it up even 48 hours while the cells were dividing. So it is definitely a long-lasting labeling effect. And therefore, we thought that this was a system that would be worth trying in cell-to-death potential for cell-based therapy, such as the human mesenchymal stem cells, which is a work that we have done with the stem cell lab of Professor Kai Kilty. So we can see that effectively the liposome ICG are uptaken. And in order to identify the working formulation that is not going to affect the cell, we performed a liposome-related toxicity assay because it is a cationic formulation, therefore it's going to interfere with the membrane of the cells. And also to look at the relative potential toxicity of ICG, which is known to have an effect on the mitochondria, we did an MTT assay, and we selected the 2 micromolar formulation to progress with our studies. And then we looked at the efficiency of the labeling of the cells, both using an in vivo fluorescent camera, as the ultimate goal is to go and label the cell and track them in vivo, but also doing a fax experiment to see the efficiency of the labeling, and we can see that the cells are efficiently labeled. So the point is that these cells are then cultured to become spheroids, and already 5 days pass by when you seed them in circular low binding plates. But you can observe that the signal remains quite strong, so mature spheroids are homogeneously labeled with the signal. And then we went to try in vivo our cells, and we performed 2 types of experiments to check the depth of penetration of the labeled cells. So we went subcutaneously, but also we tried one of the trickiest parts, which is intracranially, and we could observe that by implanting the spheroids in the brain, in the right striatum, we were able to follow the signal on the live animals up to 1 week post-implantation, and the signal was still present in all the animals 2 weeks, and for some of the animals, we were still able to detect the signal up to 3 weeks post the implantation, both subcutaneous and intracranially. So to wrap up, I wanted to share with you this information on the formulation of ICG into liposome that improves the optical properties of the ICG dye, and that our liposome ICG nanoprobes are a rapid, efficient, and long-lasting cell labeling agent that labeled MSC retained the viability and were able to form the mature spheroids, and that this then can be probably used to enable real-time, non-invasive in vivo cell imaging of transplanted spheroids that have the potential to be used as a tool to follow stem cell fate upon transplantation. And I would like to thank my lab PI, Professor Costaleros, and the whole lab, but especially the people that have directly contributed to this work, Dr. Neus Lozano and Deborah Bragaviera, our collaborators on the stem cell front, Professor Kai Kilty, and the UK Regenerative Medicine platform for funding. Thank you.
Thank you, Mariarosa.
Thanks.
Questions for Mariarosa?
I may need it.
Okay, we got an intense sessions in terms of questioning. Luisa, please.
Very nice. I have a question on the localization of your liposome field with your dyes, because I guess you changed completely the localization of the dyes in this case, because it's not any
cells,
yeah,
you mean?
it's
Yeah,
not anymore
actually,
on the
one of the
membranes
things is like, as we were saying, DIR is this hydrophobic mole with this lipid moieties that intercalate the cell membrane. And so we have a staining that is superficial on the outer layer of the cells. So one of the reasons that I think why the signal is decreasing relatively fast, is good for in vitro experiment, but faster compared to the liposome, is because as the membrane gets recycled, it's probably going to be breaking up and metabolized inside the cells. What happened with the liposomes? So IgG is an amphipathic molecule on its own, and so it gets incorporated into the membrane of the liposome. Then the liposome gets internalized, most likely by endosomal uptake. And then where that it starts the release is a release is from an inside out. And it's probably going to bind and intercalate also with the inner membranes of the internal cytosolic bodies before it reaches also on the outside. So we have a longer kind of internal release of the dye that is therefore able to be picked up for longer times. I hope this answered this question, but we can talk more about that.
No, it's very interesting, and I think that I would love to see this with gadolinium rather than a fluorescent molecule so that you can be even more quantitative. So thank you again to Mariarosa, I'm sorry, and we move to the next speaker, Dr. Dora Mann, and she will talk about analytical ultracentrifugation in the characterization of nanomedicine products. And she is from the European Union Nanocharacterization Lab. And she's based in Infra, not too far from here, and in Italy, as long as I know.
Thank you very much for the introduction. I was afraid a bit that I will be the odd one out in this session because I'm not presenting material, but I'm presenting an analytical method, what we are suggesting for the characterization of nanomedicine products. I'm coming from Infra, from the Joint Research Center, which is part of the European Nanocharacterization Laboratory, a consortium, and this work was done under the umbrella of this Horizon 2020 project. The Joint Research Center is contributing to the work of the consortium with physicochemical characterization of nanomaterials, trying to characterize nanomaterials and find these already so many times mentioned critical quality attributes, which at the end allow us to understand how the physical chemical properties will affect the interaction of these materials with the biological systems. Besides of helping academical and industrial laboratories in this preclinical characterization of their materials, the consortium is also searching for new analytical methods that could be used in the characterization of nanomedicine candidates. New methods, not in the sense of new technologies, but we are searching for technologies which are well established but could be used on this field. And analytical ultracentrifugation is an analytical technique which is something like this. It was not invented recently, it was invented in the 1920s. It had a kind of renaissance in the 70s, 80s, and it seems that since the 90s there is an increasing number of publications which include analytical ultracentrifugation, which is most probably linked also to the commercially available instruments, and this peak is maybe related to the availability of a free software for data evaluation. You can see that material science, chemistry, and chemical engineering is gaining more and more space among the publications with analytical ultracentrifugation. But how does it work? Maybe the instrument might look like a simple ultracentrifuge, but inside there is something different. You have a rotor which can spin with up to 60,000 RPM, but you load your sample in a special cell with transparent optical windows, and your sample is illuminated from the top and followed with absorbance and refractive index detecting optics from the bottom. So while your sample is spinning and your particles are sedimenting inside the sample cell, the absorption or the refractive index of your sample cell is compared to a reference cell, and this is followed in time and space. And then there is a series of partial differential equations which describe the concentration change in your cell. And as we mentioned, there is a nice software which is helping now mortals like me to solve this kind of series of equations and calculate the sedimentation speed of the particles. From the sedimentation speed, it is possible to calculate the mass of the particles, and if we know the density of the particles, the density of the liquid, the viscosity of the liquid, and the shape of the particles, we can calculate the size of our particles. How does it look like for a nanomedicine product? For a giant compared to proteins for which the method was originally developed. Our model here is Doxil, because it's commercially available in good quality and very homogeneous size distribution, monomodal size distribution, and moreover it contains an active ingredient which is absorbing at around 500 nanometer, so it can be followed with the absorption optics of the instrument. The first figure shows how does it look like when we spike the Doxil suspension with free doxorubicin hydrochloride, and we start to centrifuge in the analytical ultracentrifuge. You can see that after this signal jump of the meniscus, there are the absorption profiles which follow each other in time from the dark blue to the red, and we see that during the run there is something which is sedimenting in this sample, so the absorption is decreasing, but it never reaches the zero level, so there is always a certain background which remains and which corresponds to the free doxorubicin hydrochloride in this spiked sample. Instead, the original sample, which you can see below, contains a much, much lower amount of free doxorubicin hydrochloride according to the ratio of the sedimenting and the non-sedimenting part of the signal. It is around 2.5%. Why don't we say that this is 2.5% of free doxorubicin hydrochloride? Is because of the shift of the absorption spectrum between the free drug and the crystallized doxorubicin inside the vesicles. This value, anyway, is very close to the value which we determined with the golden standard method, HPLC, which is filtering the vesicles and determining the free drug in the filtrate by HPLC. So if there is an existing golden standard method, why do we suggest another one? It's because the full measurement with the AUC takes maybe about 1.5 hours, and instead to determine this by HPLC, to filter, to set up the right controls for the filtration device, how much it absorbs from the drug, it takes a full day. And the data coming out from AUC can be used also for other calculations. We can calculate the sedimentation coefficient distribution of the vesicles from the absorption and the interference data, and if our particles are nicely homogeneously loaded with the drug, they should match quite well, as we can see for this example. In case of a mixture, when we mixed empty particles with loaded particles, what we found that there is a mismatch between the sedimentation coefficient distribution calculated from the absorbance or the interference signal. What is surprising, that this mismatch was not so big as I expected, and also that if you just look at the sedimentation coefficient distribution of the control particles, it appears at lower sedimentation coefficient values than for the loaded particles. How can it happen that something which is according to the cryo-TM is bigger, sedimenting slower? It happens because of the difference in density. But can we measure the density of these liposomes with the analytical ultracentrifuge? Actually, yes. So we measured the sedimentation of Doxil in different concentration of sucrose solutions, and you can see that while they are still sedimenting at 1.05 gram per milliliter concentration, they are moving from the bottom of the sample cell to the meniscus when we reach 1.06 gram per milliliter. Instead, for the empty vesicles, this change happens between 1.04 and 1.05 gram per milliliter. Now, using the density, we can calculate the size distribution from the sedimentation coefficient distributions, and if we compare it with the actually used golden standard DLS, it gives very, very similar values. So then what is the advantage? The advantage becomes visible when we have a real sample, which is not so beautifully monodispersed. This is a not so good sample from our laboratory. It is daunorubicin-loaded liposomes, where you can see that even after purification, we had around 70% of free drug outside the vesicles, and that with the AUC, we were able to resolve a multimodal distribution, which was not possible to see with the DLS. So, as we have shown, analytical ultracentrifugation might be interesting in measuring size and size distribution of nanomedicine products if we know the density. But analytical ultracentrifugation actually can help also in determining the density of these materials. If we are lucky and our drug is absorbing in the visible range, like doxorubicin or curcumin, we can measure free versus encapsulated drug ratios, and we can also get a hint also about the presence of empty carrier in our product. In details, you can read more about in an article we published recently in the International Journal of Pharmaceutics on this topic. And with this, I would like to say thanks to my colleagues at the JRC, the colleagues at EUNCL, and thank you for your attention.
Thank you, Dora. Thank you, Dora, for this presentation. Open for questions, comments, curiosity, please.
Yeah, I'm glad to see that ultracentrifugation is back from where it ended up many years ago. Did you try serum concentrations or plasma concentrations of Doxil liposomes? Because that would really be an interesting approach to see how much free doxorubicin is around in these samples.
So actually, I was not really willing to show those results because maybe it is for a next publication, which is ongoing. But yes, if you do a measurement in the presence of serum, you can see the doxorubicin, which is adsorbed on the serum proteins. Moreover, you can distinguish it from the free doxorubicin, which is still in solution. And because it's an analytical ultracentrifuge, applying the right model for the sedimenting protein part, you can calculate what is the molecular mass of the protein which adsorbed the doxorubicin. So you can see that it's mainly serum albumin which is adsorbing based on the molecular mass.
Okay, this was a different question, but so because I meant, can you put doxorubicin liposomes and then ultracentrifugation and measure the free doxorubicin, but it's even more beautiful. You can measure free doxorubicin, protein bound from. Interesting. Yeah, thank you very much.
Okay. If there is Avi, yes.
Just a quick question. Do you think the liposomes can actually break under the centrifugation? Because this is a lot of force, and they are, you know, I wouldn't say they're rigid, but the membrane does have cholesterol there, so the membrane can actually break and release the medicine?
Yeah, I think it's a very relevant question. I think at this speed they were not breaking because you couldn't see an increase of the signal after they are sedimented. If they would break, then they would continuously release more and more doxorubicin. But I guess to choose the right sedimentation speed might be important. I haven't seen breaking neither at 40,000 RPM. But yes, you have to be for sure careful with this.
With the monopopulation, the percentage of free doxorubicin was very low, was like 5–6%. So
Yep.
if there is any breaking, it should be minor. But anyway, we can continue talking about that later on. So